Hello, I'm Ben Minerich, a mussel conservation specialist at the Minnesota Zoo, and I have a quick question for you. Why don't shallow water mussels understand puns? Well, it's because they're so literal. While this question hopefully merited an eye roll, I'd like to share with you how the answer to a simple question I investigated over the last 2 years has improved our mussel rearing methods at the Minnesota Zoo. But first, some background. We've been head-starting year-old mussels from the Minnesota DNR and Genoa Fish Hatchery in lakes at the Minnesota Zoo since 2015. And in 2017, we were funded to create publicly available school curriculum centered around mussels, host an annual student-led social media challenge, convert an outbuilding into a mussel interpretive center, and build a propagation research facility. I had to decide what systems to build that would offer the best growth and scientific. Up until that time, mussels had been grown in subspecies buckets, pans, and an exhibit. The last two had sand filter media for substrate and had better growth, shown in the blue and white lines, while mussels grown in the buckets on screens had reduced performance. So I chose pan systems, but one simple question remained. What size sand should I use? For mussels under 8 millimeters, fine sediments and rearing systems can increase growth rate and survival, and are proposed to act as a grinding substrate to enhance digestion. Another benefit is that bacteria on sediment and culture tanks can significantly reduce toxic nitrogen waste and are a source of food for mussels. I hypothesized that smaller sediments would aid in digestion and increase bacteria availability as food, leading to increased growth. I examined 3 sediment sizes found where my research species live: fine sand, which is what we had been using, and pebble. I also included an even mixture of all 3 to represent a more naturally heterogeneous substrate. Mussels grown in pans without substrate have been shown to exhibit reduced growth, so this treatment was not included. 3 pan systems were built with room for 36 replicates modeled after these facilities. Filtered wild water from a lake was constantly added to provide food and temperature buffering following natural thermal cycles. Sediments were seasoned in lake water for over a month before mussels were added. Mussel performance was evaluated by which sediment elicited the highest survival and growth from May 2019 to September 2020. Megan Bradley suggested I examine if the presence of bisis was correlated with better survival and growth to be used as a subacute indicator of mussel fitness. Also, I was anecdotally evaluating how each sediment impacted ease of maintenance when mussels were measured and sediment was rinsed every 2 months. Muckets were acquired from the Minnesota DNR in May of 2018, and with one growing season, they had a mean length of 31 millimeters at the beginning of the trial. Black sandshells were acquired from the DNR in May of 2019 when trials began, and had a mean starting length of 6 millimeters. That September, the minimum length of black sandshells was greater than 8 millimeters, and I tagged 24 mussels per pan. Survival was evaluated with a chi-square analysis, and percent of mussels with bisis was compared by ANOVA and logistic regression. For the muckets, there was no difference between treatments concerning survival or growth, and bisis presence did not correlate with either survival or growth. For black sandshells, there was just one mortality event at the beginning of the experiment, in which just under half of all individuals expired evenly across all treatments and pans. Spent shells recovered in July were the same size as the smallest mussels in May, and it was proposed that these individuals expired at or shortly after arrival due to transport or acclimation stress. Growth, however, was significantly influenced by the presence of fine sand, whether alone or mixed with the other sediments. Repeated measures analysis of tagged individuals after September 2019 showed that the mean of individual growth slopes per treatment continued to diverge, with mussels in the presence of fine sand having a greater growth rate. For bisis, September was the only month with significantly lower bisis presence for those reared in fine sand and sand. By January 2020, the pebble treatment had the largest percentage of mussels with bisis per pan and, as seen with the superimposed length measurements, was lagging in size. Logistic regression models supported that bisis presence was negatively related to length as a predictor for muckets, while length and treatment predicted lower bisis presence for black sandshells. As there was no correlation with time, this data suggests that the length of these species, not the age, predicted if bisis was produced. So, in conclusion, fine sand had a positive impact on growth for the black sandshells during their first and second year at the zoo, and was much easier to work with when separating mussels and sediment through sieves. So, of course, my response to this data is that I will now use fine sand in my head starting tanks. Secondly, I'd like to investigate if the composition of the fine sand was more important than the size. Sand was sourced from Wisconsin and is primarily silica, but fine sand was harvested from dry riverbeds in South Africa, which tend to have a higher clay content. So further analysis may uncover the possible impact of mineral content, angular shape, or porosity on mussel growth. I'd like to thank all my funding sources and my partners that made this work possible. Check out the description for citations, and I'm literally so excited to respond to questions in the comments section below. Thanks.